2, and 1.
Greetings and salutations. I'm Dan Kastner. I'm the Scientific Director Emeritus of the National Human Genome Research Institute, and it is my enormous pleasure to welcome you to this, the 14th installment of the 21st year of Demystifying Medicine. This is a course that has been going on now for 21 years, and our goal is to bridge the exciting developments in biology and engineering with medicine, because we know that there is so much information in these respective areas that there's more than what can fit into one head, and having a bridge, a communication between individuals that are expert in the various areas can be very, very stimulating and oftentimes lead to totally new insights. Our sessions are on Tuesday afternoons from 4 until 6 p.m. Eastern Time, January 4th through May 3rd. And they can be watched through the video cast link that is shown on the screen. Our complete course schedule can be seen by going to the link that's shown right below it. And if you attend at least 50% of the sessions and pass a not so difficult final examination, you will receive a certificate. And our CME credit number is shown here on the screen today. It is 38654. Again, that's 38654, and we will remind you of that again later on in the session. For those of you who have questions, you can submit them during the presentations by using the Send Live Feedback button on the video cast display. All of our previous sessions have been archived going back to the very beginning, to the very first year, and they're accessible at the link that you can see towards the bottom of the screen. And for additional information, you can email my colleague and, in fact, the founder of the Demystifying Medicine series, Dr. Wynn Arias, at the email address shown at the bottom of the screen. If we can have the next slide, you can see a picture taken back in the late 1800s of the iconic Brooklyn Bridge. And for those of you who are not familiar with the Brooklyn Bridge, it is a bridge that goes between the boroughs of Manhattan and Brooklyn in New York. And actually, these were separate towns at one time. And you can see on the catwalk to the left, two men who are having a discussion. One of them, I'm sure, is from Brooklyn and the other Manhattan. And they are discussing some scientific topic. Perhaps they are anticipating the possibility of endocrine disruptors in the water. And they have gotten up on the catwalk of the Brooklyn Bridge to avoid any such chemical influences. And we'll be talking a lot about that today. But in any event, certainly the communication between people with different expertise is what we are trying to foster with these sessions. If we can have the next slide, we will present information on the first of our presenters. And as is usually the case, we have at least two presenters at the session today. The title of our session is Endocrine Disruptors and Fertility. And the first presenter today will be Dr. Alan DeCherney. He is a senior investigator and deputy clinical director for academic affairs and the director of the Reproductive Endocrinology and Infertility Fellowship Program at the Eunice Kennedy Shriver National Institute of Child Health and Human Development. That's a mouthful. He got his MD from Temple University and then did an internship in internal medicine at the University of Pittsburgh and a residency in OBGYN at the University of Pennsylvania, and then was a research fellow at the Lister Institute in London. He has had leadership positions at Yale, UCLA, and Tufts over his distinguished career before coming to the NIH. He is the former editor-in-chief of Fertility and Sterility and formerly on the editorial board of the New England Journal of Medicine. In 2004, he was elected to the National Academy of Medicine. His research group studies the developing blastocyst, the trophoectoderm, and pluripotent embryonic stem cells, and investigates their interactions with the uterine endometrium at the inception of pregnancy. His group also investigates agents that affect successful and pathologic implantation at a cellular and molecular level. If we can have the next slide, I will briefly discuss our second presenter at this endocrine disruptors and fertility session. And our second presenter is Dr. Lyuba Varticovski. She is an associate scientist in the Laboratory of Receptor Biology and Gene Expression in the Center for Cancer Research at the National Cancer Institute. She got her medical degree at the University of Valle in Colombia. She did a research fellowship with Max Wintrobe and James Kushner at the University of Utah, did internal medicine residency at Albany Medical College and a hematology oncology fellowship at the New England Medical Center, and then a research fellowship with Lou Cantley in Boston, studying the PI3 kinase enzyme. And in fact, she was one of the original people working on that. She is a member of the American Society of Hematology, the American Society of Clinical Oncology, and the American Association for Cancer Research. Her research group studies mouse models for cancer and cancer stem cells. They also do work on global unbiased chromatin landscape in response to activation by glucocorticoids and other nuclear receptors in differentiation and cancer. Also work on chromatin modifications in osteogenic differentiation. And relevant to this particular presentation, they work on detecting and quantifying endocrine disruptors, contaminants in water that activate nuclear receptors affecting the ecosystem and humans. And with all that having been said, I think it's time to turn things over to the good Dr. DeCherney, who will regale us on the work of his group on this topic. Dr. DeCherney, please take it away.
Good. Can you hear me?
We can indeed.
Okay, great. So, we're going to talk from a clinical standpoint about the endocrine disruptors. This is an extremely difficult topic because you have two very variable things to study. One, human beings who are exposed to multiple toxins and non-toxins, and you have multiple toxins. So most of this is observational science and very little cause and effect. Yet there are a great number of studies that make this a fascinating topic. And this is just a panoply of the different sources of endocrine disruptors. Obviously, the pharmaceutical industry, hospitals dumping things into the waste, pesticides, and various other chemical agents. So just a personal story. This is a tanning factory where they use a tremendous amount of very potent chemicals. And a number of years ago, a chemistry professor at a renowned university came to me because his wife was a habitual aborter or recurrent pregnancy loss. She had multiple pregnancy losses, and she also had a history that her coworkers also had an inordinate number of recurrent pregnancy losses in the first trimester. So the assumption was that it was due to some of these chemicals, and although we never identified the chemical, although the husband was kind of obsessed with finding what that chemical was. We never found out what the chemical was, but we pulled those women out of the tanning factory, and in about 6 months, they were able to conceive and carry to term. So this is a good example of these disruptors as far as a clinical picture is concerned. This is something that's very, very important to the populace. They're very in tune to this. Patients are very in tune to it. Now, the majority of work we do is infertility, and there's not a lot to be said for infertility per se as far as endocrine disruptors, but certainly theoretically, since most of these endocrine disruptors turn out to be like estrogen, it probably is something that's relevant. Problem is difficult to tell. Patients are different, and the toxins are different. So there's very little in the way of cause and effect that we can pin down as far as what is actually going on with these exocrine disruptors. But the best example is an old example, first described by a gentleman, Taylor, who's a professor at Berkeley, looking at this weed killer that's an estrogen disruptor, atrazine. Diabetes, who had a very poor, in the early part of the 20th century, who had a very poor history as far as pregnancy was concerned. They aborted pretty regularly in the first or second trimester. They were given DES in order to save the pregnancy. This was actually a chemical fluke because the DES turned out to be measured as progesterone, but it wasn't. It really was just DES looking like progesterone in a radioimmune assay. But it did have long-term ramifications. The first ramification is that these offspring where the mothers were treated with DES turned out to have vaginal cancer when they were very young. And also described were changes in the Mullerian system. You can see the uterus here and a withered tube and a T-shaped uterus. So here's an endocrine disruptor that was used as a medication that caused changes incidental to increased estrogen, And this just looks at the DES passing across the placental barrier, and you can see there is some increase in infertility, but it's difficult to know whether this is due to problems with the Mullerian system or the tube, or it actually has some effect on ovarian chemistry. Another clinical example is in the male. These are males that are exposed to high doses of pesticide during pregnancy, and they have varying degrees of hypospadias. And these pesticides actually look somewhat like progesterone, pretty much like stobesterol, an open chain molecule with progesterone like receptors. And this is a study that also in humans, a kind of a cause and effect, looking at the adverse effects of endocrine disruptors on fetal testis development. This is based on phthalates, and we'll talk about phthalates in just a moment. So there is clinical evidence in reproductive organs that these endocrine disruptors really do have an effect, certainly on the fetus, and to some degree on human adults as well. And this is just an example of the problem with adults. Another case study was in patients people that worked in the operating room, anesthesiologists, surgeons, nurses who were exposed to halothane anesthetic also had a very high rate of pregnancy loss. And as soon as these halothane-like anesthetics were removed from the anesthesia armamentarium, these people went on to carry. So just another example of a disruptor having a toxic effect and an effect on patients getting pregnant. A famous study looked at the sperm count in males from 1972 to 2011 and found that the sperm count in men, this was just in Paris, went down significantly over time. Not only did the counts go down, but the abnormal morphology went up. As well. Now, this was a frightening study because it showed when you extrapolate the numbers that the male factor, the male sperm count, went down by a factor of 10% every decade. Well, that's that's pretty pretty damning. But there have been other studies, primarily the best one is the one in Seattle. It showed that this effect was not true in the United States. So this is a mixed bag, but there's no reason not to believe the Paris study that there is some effect of these toxins and estrogen itself in the water, in the Seine River, that caused patients to have decreasing sperm counts. And earlier we talked about declining fertility rate. There is a declining fertility rate around the world, as you can see here on this slide, perhaps exacerbated by the virus, but nevertheless, this is going down. Now, of course, it's hard to attribute this to toxins. A lot of this is socioeconomic as well, but nevertheless, there is a question that as the toxins go up and people are exposed to more neurotoxins, as chemicals in the environment continue to increase as waste products of the industrial complex, then perhaps there is something to be said. This is one of the causes of a declining fertility. So some of the ways that it works, these disruptors work, metabolic disorders, hormonally related disorders such as increasing cancer, thyroid changes are an old example with patients developing goiters based on iodine, lack of iodine, and of course, most important and most misunderstood are immune factors. Now, these are some of the agents that we're talking about. The phthalates, as you can see, are a form of plastic, have vinyl floor, food preservatives, non-steroidal estrogens, as we talked about, DES, the polychlorinated things that are closed in plastics and in insecticides. And the one that has the most, as you'll hear about, the most that has the most play because there was a lot of publicity, primarily in plastic bottles, and are considered a contaminant. Another association is dioxin. Dioxin is supposedly associated with a high degree of patients exposed to a high degree of dioxin will develop endometriosis, and that seems to be documented. So I don't want to go, I'm not going to delve on this slide. You can just see a list on the left of the different agents that we're concerned with, and I've mentioned pretty much what occurs as far as aberrations in these with utilizing or people that are exposed to these various agents. DES we mentioned as far as the tube is concerned. Insecticides we mentioned as far as the male frogs were concerned, just as an example. And how do these work? What is the mechanism of action of these estrogen disruptors or any hormonal disruptor? Anything that looks like these are partial ligands. They look like estrogen. They look like progesterone. They look like cortisone, but yet they're different. But they have enough similarity to the active agent, the active hormone, that it interacts with the receptor and gives the receptor kind of the wrong message, so that the message that turns out is not the normal message, but a message of too much aromatase, or not as much aromatase. And you can see here in this graph, you can see the key characteristics of estrogen disruptors. You can see receptor expression increased, receptor expression decreased, change in Ligon, as you can see at the very top. And of course, it also causes, these also cause increased production of of hormones, or decreased production, or production of abnormal hormones as well. So just from a clinical standpoint, just to mention a few of these, biphenyls are in plastic. You can see the plastic bottle, and there'll be some mention about what happens as far as these biphenyls are concerned in regards to patient and consumer pressure. But the evidence is pretty good, at least in animal models, that this does cause some changes in ovarian morphology and abnormal folliculogenesis. And exposure is somewhat elevated in patients that are infertile, but like I said, this is hard. This is so hard to tell, to separate social, economic, other health aspects from aspects from the toxins themselves. There's a study that was done in San Francisco at UCSF that looked at some of these toxins as far as infertility was concerned. And although they were able to come up with some paradigms, still these were more hypothetical than they were proven. And the same thing in the male. You can see that decreased sperm counts and motility are influenced by the biphenyls, yet the studies are inconclusive because the male sperm count changes normally anyway, so it's difficult to actually document. The phthalates are another plastic byproduct. You can see that they're in children's toys. And there also are some studies as far as impaired fertility is concerned. But once again, I don't have slides to show those studies because the findings are hypothetical and not strongly documented. As you can see, few and underpowered. So pesticides, another that we mentioned, especially the ones with the male frogs, also act as hormones. So they're toxic, but toxic causing necrosis, but they're also toxic as far as acting like hormones and activating receptors that cause the cell to behave in a non-predictable and non-programmed way. Phytoestrogens are estrogens that look very much like estrogen. They're found in vegetables, fruits, plants. In fact, there was a time that people thought that this was a good way to treat menopause with these phytoestrogens. But it turns out the phytoestrogens did not respond and perform normally. Actually, they are a toxin, and they do stimulate the estrogen receptors, both alpha and beta, to perform in aberrant ways rather than in healthy ways. So these are not used anymore as a therapeutic, and actually people are cautious that they can perform as a toxin. And you can see here they stimulate progesterone and estrogen production, so patients become hyperestrogenic, and they can also cause a premature puberty in females, and they can suppress spermatogenesis in the male. And the problem with these estrogen disruptors, we're all exposed to them at different levels, so it is a ubiquitous threat as far as exposure is concerned. But nevertheless, it is a fact of reality, and how profound it is difficult to tell, and we'll hear from that when Lyuba gives her talks. Now, the country is interested the FDA banned BPA. The phthalates are monitored by the Consumer Protection Safety Commission. The child care toy industry is monitored heavily as far as this is concerned, as well as cosmetics where they're used. So the public is aware, and the regulatory agencies are aware of these and the damage they have as far as clinical factors are concerned. But once again, it's hard to pin down because it's not a direct cause and effect that can be extracted from this. And pesticides too are highly regulated. The phytoestrogens are unregulated. They still carry the mantle of being healthy because they act like estrogen, but they're not a true estrogen, and actually have a deleterious effect. So this is my last slide. It's a summary slide. It just looks at humans, rodents, and zebrafish. It looks at some of the studies that are done. We talked about sperm quality going down, both in number and in its morphology. Fertility is difficult to document, but more important is the fact that recurrent pregnancy loss can be documented as exposure to toxins. And of course, the DES story, as far as cancer is concerned, is well known and well documented. Very little is known about implantation, and so it's difficult to know. Of course, we do in vitro fertilization, and many feel that there are contaminants in the media that we grow our embryos in, and perhaps that accounts for less than 100% pregnancy rate. And the studies in rodents and zebrafish, although it's cause and effect, do document or enhance the findings in the human. It's not close enough to draw permanent conclusions. So in conclusion, estrogen disruptors exposure is unavoidable. Human studies show mixed results for ecological changes. DES is clearly harmful. That's an old one. And this stuff was done. DES was used in the 50s. BPA has the most evidence for reduced fertility, and I'm not sure that's not due to most interest and people taking up that cause. And in the future, science, I think, will move ahead where we understand how the receptors respond and perhaps be able to call out what are the dangerous endocrine disruptors and which are not as profound. So on that note, I'll stop, and thank you for your attention.
Well, thank you so much, Dr. DeCherney, for that really fantastic overview. And what we're going to do is to go on to Dr. Varticovski. And if you have questions that are already in your mind, that are burning questions, just put them into the question box for the video cast, and we will tabulate all of them. And then at the end of Dr. Varticovski's talk, we will have the Q&A for both Dr. DeCherney and Dr. Varticovski. So with that having been said, I think that we're ready for Dr. Varticovski. Lyuba, please take it away.
Thank you very much for your introduction, and I am really very pleased to be here today and present some of our work on the endocrine disruptors and share with you some of our concerns about fertility. And so my talk will be on endocrine disruptors and sex hormones. I have nothing to disclose. Perhaps next time. And today we will go over the definitions of EDCs, why does it matter, sex hormones and other nuclear receptors, consequence of the disruptions, hormonal mixtures and how we deal with that, and a very interesting mechanism which has been working in our laboratory on assisted loading between nuclear receptors. So we go a little bit deeper into what are the endocrine system itself. We have a number of endocrine organs in our body, including the brain of hypothalamus and pituitary, parathyroid, thyroid, adrenals, pancreas, ovaries, and testes, of course, male and female. And endocrine disruptor chemicals have been defined by WHO in 2012 and redefined again a few years ago. And it really is a mixture or any exogenous substance that alters any function of the endocrine system and cause adverse health effects. So that can be on intact organism, on the progeny, or its subpopulation. There are some very important features of EDCs that I will go over. And one of it is effect at low dose. The high dose effects might be different, like what we see peak hormone effects during pregnancy. But what we see with EDCs, it's what we call a non-monotonic effect, which means they not necessarily correlate with the concentration. They disrupt the normal flow of endocrine system. So they have very wide range of effects because the endocrine system is evolved to respond to very low levels and very small changes of hormones, the regulation of the synthesis of each other and variety of other hormonal and non-hormonal growth factors and differentiation factors, and therefore makes them that they really regulate or can disrupt signaling that governs all tissues and all organs. They are persistent and they have some of them very latent effects, as we heard from the previous talk by Alan, the attorney, such as dioxin, bisphenol A, and phthalates. And they certainly ubiquitously in the nature today. So the increasing evidence suggests that their detrimental effects on health and wildlife, humans, including reproductive health, on our entire ecosystem. So why does it matter? The endocrine system, as I mentioned, can be disrupted by even smaller gestures. And EDCs are mimicking natural hormones. And I'll give you some of very specific examples of how that mimicking can be so difficult to study. And they can be having biological effects that have been documented for many contaminants. And of course, humans are continuously exposed to endocrine disruptors in every form and shape possible. Most importantly, that the vulnerable period in both animal and fish and humans are in fetal development and early infants, because that's where the whole organism is tightly regulated for growth and differentiation, and also, of course, can be effective in later in life, and those effects may persist for later. The other issue I want to point out is that mixture of several classes of endocrine disruptors are very, very frequent. So what about chemicals? Well, we have a stockpile of chemicals that tripled over the last decades. And their regulation is based on a risk assessment of individual compounds. But in real life, we will never be exposed to individual compounds. This is a mixed bag. So we have CAS registered chemicals all over the world. And this is a very extensive study published just a few years ago that is near 250,000 compounds have been registered that are currently in production. There are other registries, like some of the Canadian and European separate registry contains another about 70,000. And in addition to that, over 50,000 compounds are not, we don't know the structure because they're proprietary by industry and they're protected for patents and so on. That makes it even more complicated. So let's go over sex hormones. We think about sex hormones perhaps in general terms, but we're going to focus on more specifics of how actually it happens. For our laboratory, it started at the turn of this century because there were reports that fish floating in Potomac River were strange. They were killed, they had ulcer, they were stressed. And I want to focus on this very bottom of the slides, that the biological examination of their organ, various organs, found very unexpected findings. And the findings was that this fish had intersex. So that not only depressed, they had been exposed to both male fish to female hormones and female fish to male hormones. So this fish had intersex organs. And then there became an avalanche of early studies of why the water is dirty. And here is a cartoon, says, face it, Bill, you have changed. And the Bill obviously looked like female. And that was very strange. And the avalanche of scientific reports and newspaper articles on intersex presence of fish only in the Potomac. This is limited to our river floating right in front of our eyes. So let's go over some of the characteristics and regulation of sex hormones. So hypothalamus and anterior pituitary regulate the production of precursors of the sex hormones. Gonadotropin releasing hormones released by hypothalamus regulates production of follicular stimulating hormones, luteinizing hormone, and ACTH. And these are produced equally between male and female. So we have sufficient grounds to think that any of this disruption will affect both sexes. Well, then the adrenals come into play and they produce progesterone. And when we think about progesterones, we think about pregnancy, we think about ovulation, anticonceptive therapy, all kinds of things, but that's not the case. Progestins are actually the building blocks of androgens. So without progesterones, we cannot have androgens. And androgens, which are primarily made by the testes, but also made in other organs, as well as in the females, make estrogen. So they are the building blocks for estrogens. So here's a delicate balance where any of these disruptions can cause dysregulation of gonads, peripheral tissues, bone formation, and metabolism. And of course, mostly in the male, normally regulating spermatogenesis, and in the female, developing female organs, as well as pregnancy and embryogenesis. So we have the three main sex hormones that we will talk about. So progesterone, androgens, and estrogen, and they act via their receptors. Yeah, I'm not going to go into detail of which one of them and how many receptors. We're going to generically consider the receptors as being their emissaries, the working horses. So androgen receptor, estrogen receptor, and progesterone receptors are only a subfamily of a number of nuclear receptors. There's 48 known nuclear receptors. And they have similarities in structure, and they have a dynamic function. And I tell you how that is so important. The structure contains ligand binding domain, which initiate conformational changes. In these conformational changes, regulate nuclear translocation domain and activation domain. So a lot of those receptors are always normally in the cytoplasm, and only in response to the hormone, they go into the nucleus. And here is schematic representation. When the hormone itself travels to the cytoplasm, where the receptor is bound and hidden by usually a heat shock protein complex. Upon binding to the ligand, this complex dissociates allowing the receptor to either dimerize or trimerize, and sometimes into quadruplicate structures, and this travel to the nucleus. And that's where they do their effect, because they have DNA binding domain, which now is allowing the hormone receptor to bind to DNA and initiate gene expression. So important feature of these particular receptors is that the activated nuclear receptors become transcription factors. And that is very important. There's no other intermediate, but they do interact with other co-receptors. They interact with different chromatin remodelers to be able to form proper structure and initiate gene expression or block gene expression. So why does it matter? For us, EDC interfere with normal function of the endocrine system and therefore can harm every organ in the living organism. They're especially dangerous, as I mentioned, in early life, and they're present everywhere. So how we protect our infants and our lives? And we need public awareness. Public awareness for us is essential because the presence of EDC in the environment at the moment is not sufficiently regulated. The screening methods not always include a biological readout, and I'll go over that very important point. For example, in fact, so much literature, so much work have been done on BPA, as Alan DeCherney pointed out, that BPA was removed from bottles, from 91% of bottles for infants, only after a big public outcry. Unfortunately, all of us have BPA in our bodies, and I will show you the data for that. So here is BPA. I'm not going to dwell too much on BPA, but I want to tell you that it is detected in 93% of over 2,500 urine samples from people who are 6 years and older, and is the highest production volume chemical in manufacturing of any kind of polycarbonate, epoxy resin, and everywhere else. And so its half-life is short, but we're exposed to that continuously. It was originally synthesized as estrogenic compound, and it does have a potent estrogenic effect. However, it also activates pregnane receptor that regulate drug metabolism and estrogen-related receptor. It's a potent antagonist of androgen receptor and thyroid receptor. So here we go. Estrogenic and androgenic effects block effect of estrogen on human body. It is diabetogenic and induces insulin resistance, type 2 diabetes, and possibly linked to other cardiovascular diseases. Furthermore, effect of BPA on androgen and thyroid receptor are transgenerational. These do not induce DNA damage. They are epigenetic. Nevertheless, they are transmitted to further progeny. So why does it matter for these compounds? And these are PFAs. We have over 5,000 PFAs in common products like Teflon pans, food packaging, waterproof jackets, carpet, and of course in the dust, because that's what it comes from, the carpets. PFAs are extremely persistent with a half-life to up to 8 years. And antagonizing thyroid receptor, we know that thyroid receptor inhibition and decreased thyroid production in the during development can lead to cretinism and really severe mental defects. And in fact, no human studies can accurately detect and quantify all PFAs because we just don't have been focusing enough technology to detect them. PCB, which is polycarbonated bisphenols, I think Alan went over that as well. These include dioxin. Dioxin has been banned in the United States since 1978, but the derivatives and all the PVC are present everywhere, soil in soil and air, and they accumulate in fish fat. So fish that floating in the river and drink the water, and water that irrigates our fields. And they also transmit it in the breast milk from the mothers. It affects neurodevelopment. Higher doses of dioxin have been found to induce genital malformation, infertility, and abnormality in sperm, as we pointed out earlier. It's also a possible carcinogen. And phthalate as PVCs, and I think Alan spent some time talking about them. So I have one slide on male fertility and how evolution of sperm quality has declined, and I think we talked about already. And this is the data from that study from France. How about neurodevelopment? A very important and interesting point of a study that has been published in 2017 from Brown University. They're looking at 2 million population, 1 million population of presumably exposed individuals that have lived in the area and have measurable in urine or in blood endocrine disruptors. That is exposed individuals and unexposed, which is in blue. You may not see that very well, but the dotted line is the exposed population. Perhaps they're very close. However, if you look at this gray line right here, it separates people who have IQ under 70. That is severe disability, and in exposed population is twice as high as it is in unexposed population. There's a clearly effect in neurodevelopment. This is atrazine, and this is another slide complementing some of the work that Alan presented. And highly agricultural state have a very, very high atrazine concentration in drinking water. So I would say that the burden of the disease, and very recently, 2015, calculated 268 million disability adjusted life years and over 9 million premature deaths each year. And it is 3 times as much as we lose people from HIV, tuberculosis, and malaria combined. It is much greater burden in low and middle economy income countries. And that brings me to another point that is very important, that higher exposure of poor, minority, and disenfranchised populations to endocrine disruptors, to toxic chemicals, to contaminated air, water, unsafe working place, and it results in disproportional health decline in these populations. We cannot blame only this to their health decline, but it is clearly a very important factor which is very frequently overlooked. I'll give you some examples. EDCs and BPA and phthalates are being much higher exposure in Latino and Black population. Those we can measure in the urine and blood. Hispanic farm workers in California are exposed to organophosphate pesticides, such as parathion, and other EDCs linked to sex dysfunction. They have much higher rate of endometriosis, fertility problems, all of these things. And organophosphate pesticides are the larger contributor in costs associated with EDC exposure. It estimated of $121 billion in the European Union and $42 billion in the United States per year. And also increased cancer rate. Cancer rate in testicular cancer of children exposed in utero to androgens, and they are higher in residents and people who work in cement factories and lime and plaster plants. So that brings me to the main focus of my talk, which is how do we detect EDC? And I'm going to give you some of the history of detection of what is the novel developments in that field. So we started in the early 80s when the endocrine disruptors came to life as a potential problem in our society and progress. And cell proliferation assays, cell count, and detecting live dyes were very early in the 1980s. Lots of publication that. It evolved by liquid chromatography, purifying some of the fractions that had effect. And then radioimmunoassays, the antibodies came to life. Then we started to manipulate the genome, and we were able to make reporter assays in the 1990s, and following by transcriptional activation assay, which are more sensitive, but nevertheless, all of them quite laborious by 2000. And more recently, the very sophisticated high pressure liquid chromatography combined with liquid chromatography and mass spec to detect single and multiple hormone analytical methods, which are very complicated, very laborious, and high cost. The only problem with that is it's a huge investment, but they have to identify compounds that have already known chemical structures. We have to be able to compare them with what we have in the library to know what does it look like. Otherwise, it is of no use. So here we are. We now 2010, more ligand binding assay on multiple targets have been developed, and those are much more useful to do somewhat larger screens, although they take a long time. They're still quite laborious. So what we really needed is a very high throughput screening on single and multiple receptors. And one of them was developed in our laboratory, and I go with that for you in great detail. So we know that the hormone, when it binds to the receptor, induces the receptor to go into the nucleus. Our laboratory have been studying nuclear receptors for a long, long time. And we had constructs where we can follow the receptors linked to fluorescent dyes, like GFP, YFP, or any other dye. So here is estrogen receptor. Estrogen receptor is naturally sort of shuttles between nucleus and the cytoplasm. It's mostly in the nucleus. You can't really see it going into the nucleus. Glucocorticoid receptor, on the other hand, is really naturally sequestered in the cytoplasm. And this is the perfect mechanism by which we studied glucocorticoid receptor. So by binding to the ligand, it quickly goes into the nucleus. And I'll show you some examples. So we can make a chimera. We take a piece of glucocorticoid receptor that will sequester estrogen receptor in the cytoplasm. And then binding to this proper ligand of estrogen to its ligand binding domain, it induces translocation to the nucleus. And that way we can make many hormones just being clearly and cleanly sequestered in the cytoplasm. So expression of this chimeric receptor, nuclear localization, selection of stable cell line, mammalian cell line, human or mouse cell line. We tend to do everything in human cell line, picomolar, nanomolar sensitivity, and it's very fast, take 30 minutes to hours. Depends on the translocation speed. And most importantly, there's no need to identify the structure of the ligand. We don't really care because if it activates the receptor, it's dangerous. So we did some of the studies. I'll go over some of the studies with you for screening for androgen receptor and the glucocorticoid receptor. This was the first study in the United States that detected glucocorticoid receptor in water. And these are river water. Water was given to us by US Geological Survey. They collected it over several years. We concentrated the samples and calculated the actual compound activity based on its original presence. And this is a schematic representation of this filter that they put at the bottom of the river. So it either grab sample or the filter, and turns out the results were quite similar. So this is how it's done. Take the water from the river, concentrate, and then put it in the cell. And if the cell is reacting with some endocrine disruptor, it will be translocated to the nucleus, and we have an opera system which calculates the ratio of the dye of fluorescence between nucleus and the cytoplasm, and therefore give us a quantitative report. So this is pretty much the plate. This is a 96-well plate that we used in the past, and now we use 364 plate, much faster. It takes more compounds, and we do dilution of various natural hormones, and then we put the samples that are unknown activity. So this is how it looks. You can see that glucocorticoid receptor linked to GFP. You see the punched out nuclei by staining with DAPI. You see the nuclei themselves, and this is an overlay. And when we add the hormone, we see just the nucleus. And here is again DAPI stain and an overlay. So this is clearly a translocation. We see nothing in the cytoplasm, a very clear and very clean assay. So this is the results from map of the samples that we collected in the eastern corridor of the United States. That was our first study. And in this study, we screened for glucocorticoid receptor activation and androgenic activity, activation of androgen responses. And here is only a few samples that were negative. They're in green, and they're negative for either filter or what we call the grab sample. And black is positive for either filter or the grab sample. And I want to point out that the red triangle and the circles are the most common samples. Therefore, these had both androgen receptor activity as well as glucocorticoid receptor activity. One of them was very high. It was not far from us, was in the upper part of Maryland. And we asked our collaborators, we had a sample which was collected 5 years ago. Could we please ask them to go back to that the same part, exactly the same spot in the river, and collect for us another grab sample, because this initial sample came from the filter. Just give us a little water so we have enough. We wanted to identify what is really the source of that activity, of those activities, because they were both. So here's how it looked. I don't have it in green. The machine sees it in black and white. This is glucocorticoid receptor activity. And this is dilution, concentrated sample 10x, 20x, 50x, 100x, and it compared with a very strong hormonal effect of the dexamethasone. And the same thing from the androgen receptor, compared with the testosterone. So activity of the glucocorticoid is quantified in the bar graph, and you can see that even 1x is already activating and detected as activity, and it also detected gene expression. So without concentrating the sample, we already see that there is gene expression alterations, which means that anywhere in the fish and the plants, where in the natural fat components, these endocrine disruptors, sterols, will naturally accumulate. They will have 10x, 20x activities. Same thing with the androgen activity and gene expression. So this sample, 5 years later, we were able to do some very sophisticated analysis by HPLC, mass spec, trying to identify it. And in HPLC, peaks 1, 2, and 3 had activity. And in the database from 2010, they were similar to known androgenic substances. And this is a result from mass spec. And this was done by Paul Klausmeier and our collaborator from Frederick. However, out of all that work, for about a year, that we did with this sample, with all the sophisticated technology that we have, we could not identify any glucocorticoid structure that we know. Well, that means the biological properties of that endocrine disruptor have changed, and it is not present in our libraries. This is a second study I want to share with you out of many that we have done. And this is on Rappahannock and Mattapony rivers in Virginia. Rappahannock River borders between Maryland and Virginia. It flows into Chicopee Bay, and of course carries all of these endocrine disruptors with it. It's a beautiful river. Here is a bridge over, many of you know this bridge, over the Fredericksburg, and it's certainly a beautiful river. Unfortunately, it's highly contaminated. Our study found that almost all samples had at least one activity, and most of them had a mixture of activities. 80% of samples had androgen receptor activating properties, and nearly 70% had aryl hydrocarbon activity. And half of them had glucocorticoid activity, and 30–40% had thyroid receptor activity. So many samples had multiple activities. So the conclusion of some of this study, it's a widespread contamination and structural changes that are induced by biological, chemical processes, algae, microbes, who knows what's present, temperature-dependent changes, and they convert these contaminants into invisible weapons. They cannot be identified by chemical structure. So there are many of them are mixture, and high throughput screening is very important and can be used for EDC screening. Well, we were talking about single hormones. Let's talk about a mixing hormone. So what happened about a mixture? Do they act independently? Do they cooperate? Do they antagonize each other? How do we know? So say two hormones. We try combining just two. And this is a ChIP-seq result from our laboratory, where you're looking at glucocorticoid receptor activity in response to its potent hormone dexamethasone, or dexamethasone followed by estrogen in the media for the cells. And you can see that there suddenly we see appearance, new sites at the DNA that binds glucocorticoid receptor, because we're chipping for glucocorticoid receptor and we see that they are extra. And some of them disappear. So they are both activating as well as suppressing some of the sites that normally glucocorticoid receptor bind. The other way around, when we look at estrogen receptor alone has certain pattern of binding to DNA. But when it combined with dexamethasone, it suddenly has other properties. It binds to a whole bunch of other sites that is not present when it's alone. Here is another site as well that probably comes from glucocorticoid itself. And this is a third one that neither one of them had before. So there is a very important modification that occur when two hormones present in the same mixture. So here we go. Schematically, when we have a hormone that goes into the nucleus, a hormonal receptor most likely finds inaccessible chromatin. The nucleosomes are closed. It may require another hormone to remodel that particular site, so the hormone receptor can actually access it. And we know that increasing evidence suggests that multiple steroid receptors contribute to progression of breast cancer. And now we know the same for other endocrine cancer, ovarian, endometrial, and prostate cancer. So this is this old static concept, for example, of FOXA1. For those of you who know, their FOXA1 has been considered an important component of estrogen receptor activity. But however, what we found that progesterone is one of the hormones that modifies, that opens the chromatin moves away the nucleosomes and allows FOXA1 to interact more efficiently. In some cases, without it, it won't interact at all with DNA to initiate gene expression. So this underscores the complexity of establishing guidelines for what we call acceptable levels of EDC, especially because we know that the majority of them are present in the mixture. So where the EDCs in the water come from? They come from runoff from natural and added hormones from cows, livestock farms, in agricultural fields from fertilizer, herbicides, and animal food additives like progesterone is frequently added to enhance growth of animals. Industrial plants, cement, plastic, paper mills, hospital, domestic waste, and of course, drug development in drug industry. Metals also modulate the function of steroid receptor. Cadmium is a very potent estrogenic effect because it acts on some of the estrogen receptor co-receptors. And so it's required almost for effect of the estrogen. And essential elements also are estrogenic, such as copper and zinc. And some people take a lot of zinc, so I don't know how that works. Anyways, we have sewage treatment plants that work now with chemical filters. Chemical filters do not completely remove sterols. Sterols have that very special formula that I showed you in a couple of slides, and they're very difficult to remove. Activated charcoal is probably a most effective way to remove sterols, but it's very expensive. And of course, disposal is an issue. Charcoal has been taken entirely out of the sewer plants treatments. So natural hormones are highly expressed during pregnancy and menstrual cycle. It's exerted by humans, by livestock, and by other vertebrates. I'll give you one example. Farm animals in US alone excrete about 49 tons of estrogen in 2008. And I'm sure that now that we have more farm animals, they excrete more. Another example calculated in China, it is estimated 3,000 tons per year of sterols are released into environment. And they only marginally reduced by chemical and established waste treatments to about 2,000 tons per year. I can give you a long list of examples, but I'm going to stop there and tell you that nobody wants to see a sign that says, warning, contaminated fish. And of course, fish are not happy as well. And we almost feel like the Sisyphus pushing the rock up the hill because it's going to roll back again. But we need to roll it up. We need to have awareness. We need to put stop on uncontrolled disposal and production of chemicals. And we need to keep an eye on what it does on human population as well as our ecosystem. So this is our wonderful group under skillful direction of our branch chief and principal investigator of our laboratory, Gordon Hager, my closest collaborator, Diana Stavrieva, and Razi, who has done a lot of hands-on work with me, and many collaborators from US Geological Survey, from environmental sciences at NIH, EPA, FDA, Institute of the Translational Scientists at NIH, and the DCEG at NCI. So I'm going to stop there and take as many questions as I can in the remaining time. Thank you.
Well, thank you, Dr. Varticovski, for that very, very clear and interesting presentation. I must say that the combination of the two presentations this afternoon is troubling to some of us, that's for sure. And just seeing some of the torrent of questions that are coming into the chat box, I think that our audience is also their minds are in overdrive right now with regard to some of the possibilities here. So I have one question that came in a while ago that actually either of you might want to answer. And this question is from Veronica, and it's as follows. Is there an impact from estrogens excreted in urine from oral birth control pills? Does it get into the water supply? Is it bioaccumulating throughout the years? I think probably both of you will have comments about that.
Can you hear me?
Yes, we
Yes.
can. Mhm.
Okay, great. Yes, it does get into the water. It's not a pure estrogen. It's actually a modified estrogen. It's not pure estrogen. It does accumulate in the water. Now, not much is, it's a very interesting question. Pill has been on the market for 60 years and it's used throughout the world. But I don't know any data where that specifically has been looked at. A problem is that it's because of it's modified, it's a difficult assay. If you take a patient on the birth control pills and do a blood test and measure estrogen, you won't pick up the estrogen that she's on. So it's a difficult assay, but definitely it's a fair question. It definitely is accumulating and it definitely is excreted in the urine. It's not changed. It just gets changed into something that's water soluble. And then gets excreted in the urine. So it's an interesting question, but I don't have any data as to what effect it has. It does get through in the breast milk. That has been shown. That's why it's recommended that patients don't use birth control pills until they're finished breastfeeding.
Well, I can add that the birth control pills are only one of the drugs that also getting flushed down the toilet. And there's a very extensive programs for drugs that have not been used to be collected and returned back to the pharmacy. They go to a special waste disposal. And a lot of people, when they have, they take the quick pack of dexamethasone or other hormones when they have allergy or something, they just flush it down the toilet. That is very, very dangerous because it ends up in our waters and rivers and lakes.
Indeed, yes. And these are very, very important questions. Well, I have a second question here. Are there evolutionary theories to explain the ease at which these disruptors can alter fish and amphibian gender? The gender of some turtles, too, seem to be influenced by egg temperature. So the question just has to do with, are there certain species that seem to be more sensitive to some of these effects than others?
Well, that's true. There are species that are more susceptible. I mean, the male frog, as I mentioned, but I think that it's not evolutionary. I think it's counter-evolutionary.
Yes. Yeah.
Yes, I think there's a lot of work is done comparing different kinds of fish to the same doses of endocrine disruptors, and some species are clearly more sensitive. And also to point out that fish specifically and amphibians have a very short window of time during their development where they all actually differentiate into their sex. And so if the male fish in that particular window is exposed to female, either progesterone or estrogens, it will develop both organs, and therefore it's infertile.
Mm-hmm.
I mean, I think the question comes from there are breeds of animals that are bisexual and do change their sex throughout their lives, and that is evolutionary. It's to preserve the animal phylogeny. So I think that's true, but I don't think that the fact that these disruptors enact that, I don't think that's evolutionary.
Of course, yes. Okay, here's another question. Someone is asking about these disruptor chemicals in tap water and in bottled water. And is it removed by water filtration plants? This is obviously a very practical question. What do you guys have to say about that?
Well, it's very interesting. We tested about 30 currently existing different production of bottled water. We did not detect any endocrine disruptors in any of them. So bottled water seemed to be pretty clean. So far, so good. We may have to repeat that study again at some point, taking up again the same and new water producing. So these are already filtered multiple times, and they come from mostly from springs and water that is being already tested to have very low level of metals and all of the other components. So we have tested tap water. Tap water periodically has some aryl hydrocarbon receptor activators. And we're not sure if that is from copper lining or some of the other components of the lining itself. But by and large, the bottled water and the tap water are clean from the contaminants. At least what we tested is Maryland, Frederick, and some of our students who live in Virginia.
Well, if that's true, then why do these agents represent a threat if they're filtered out in the water? That means we're not exposed to them.
People are exposed to them because we eat fish that drink the water that is contaminated. The contaminants precisely accumulate in fat in fish of both algae, plants, and so the whole ecosystem is the one that feeds us. So we are exposed through use of chemicals at home, plastic bottle, plasticizers, and so on, carpets, jackets, and everything that has all the other things. In addition, we eat products, no matter where they come from, the water is contaminated. So even the cleanest of farms, organic farms, still get the influence of the same water. So we're in trouble for that side.
All right. Well, following up on that particular comment, one of our questioners must have mental telepathy, I guess. They are asking the question of whether we should purchase wild-caught fish at the fish market or farm-raised fish.
I'm not sure what's the question.
So in other words, you know, if you go to certain fish markets, you can either buy fish that are caught, you know, out in the ocean or you know, in the lake or streams or whatever, or there are some fish that are raised on fish farms. And the questioner wants to know, should we prefer one over the other?
Well, freshwater fish, it probably doesn't matter because caught farm-raised fish are really on a part of a lake or part of the river, isolated, and fish actually are fed sometimes forcefully and sometimes get additional hormones. They're fed corn, they fed other additives, which might be pollutant. So they are not more protected than wild fish that's floating in the river. Now, if I have a choice of getting a farm fish or ocean caught fish, I probably go for the ocean because at least we don't know how much endocrine disruptors are in the ocean because we can't measure them in salt water, but I guess they're a little more diluted. So on that side, I don't think it makes any difference if there are fish raised in a farm or in a wild. It really doesn't.
Mm-hmm. And so there's a problem, you're saying, with detection of the disruptors in salt water. It somehow interferes with the assays?
We could not so far remove salt without removing the actual sterol. So that's a very complicated process. So most of the measurement in the soil, air, and water, what we call water, is done in freshwater samples. And there are a couple of studies that have done it in oceans, but mostly in the fish near certain vicinity. For example, there's one very interesting study in China, where is an island where people are mostly are eating fish, and the fish comes from the surrounding ocean, and their content of endocrine disruptor was extremely high. So, and sterols, and you know, we think it's a problem for people who are base their diet on marine organisms, because that